Hello students, welcome to our class. Today's portion is about enzymes. Okay, it is a common term for you, enzymes. What are enzymes? Please tell me, what are enzymes? Okay, last year we studied about different enzymes in the digestive system and all. Okay, amylase, it is an enzyme. Proteases, pepsin, renin, these all are different type of enzymes. What are enzymes? The enzymes are biological catalyst. Catalyst means something helpful for chemical reactions. So, biological catalyst helpful for the chemical reactions in the living system. Clear? So, the enzymes are biological catalyst promoting chemical reactions in the living system, and these enzymes, they speed up the chemical reactions taking place in our body. Digestion. They speed up the process of digestion. Okay. So, helpful for the conversion of carbohydrate to glucose with the help of enzymes. So, they speed up the chemical reactions or the biological reactions. So, enzymes are biological catalyst promoting chemical reactions in the living system, and we can say that the enzymes, they speed up the process of biological reactions taking place in our body. Okay, and the substances on which an enzyme acts, and we call that substances as substrate. Okay, എവിടെയാണോ ഈ enzyme act ചെയ്യുന്നത്, ഏത് substanceലാണോ, we call that substance as substrate. Okay, the substances on which an enzyme act is called substrate, and after the reaction we will get one product now. We call it as end product. Okay, so the enzymes are biological catalysts promoting chemical reactions. They are biological catalysts. They promote chemical reactions that is taking place in our body. And the substances on which the enzymes act is called a substrate, and the end product that is formed as a result of this chemical reaction is called either, for the final product we call it as end product. Okay, next about the different types of enzymes. Next about the classification of enzymes. So, International Union of Biochemistry, IUB, International Union of Biochemistry classified enzymes into different groups. So, what are enzymes? They speed up the chemical reactions in the living system. So, this IUB, that is International Union of Biochemistry, they classified the enzymes into different classes. First one, oxidoreductase. Oxidoreductase, helpful for the oxidation reduction reaction. Okay, pyruvate dehydrogenase that we studied, it is an example for oxidoreductase, helpful for the oxidation reduction reaction. Then transferases, the transfer of specific group from one substrate to other. Specific groups, you know, carboxyl group, acid group, alcoholic group, that you already studied in your chemistry. Okay, OH group, COH group. അപ്പോൾ ഏതെങ്കിലും ഒരു സ്പെസിഫിക് ഗ്രൂപ്പിൻ്റെ അങ്ങോട്ടും ഇങ്ങോട്ടുമുള്ള ട്രാൻസ്ഫർ, ദാറ്റ് ഈസ് ട്രാൻസ്ഫറേസസ്. ട്രാൻസ്ഫർ ഓഫ് സ്പെസിഫിക് ഗ്രൂപ്പ് ഫ്രം വൺ സബ്സ്ട്രേറ്റ് ടു അനദർ. നെക്സ്റ്റ് വേർഡ് hydrolysis. It is a hydrolytic enzyme that helpful for breaking down of complex compounds into similar compounds or similar substances. Complex substances into similar one. Lyases, that catalyzes the removal of a group. Helpful for the removal of a group. Group think അറിയാം, different groups that you studied. Okay, next one, isomerases, that helpful for isomerization reaction. ഒരു കോമ്പൗണ്ടിനെ വേറൊരു കെമിക്കൽ ഫോം, സ്ട്രക്ചറൽ ഫോമുല ഉള്ള കോമ്പൗണ്ട് ആയിട്ട് മാറ്റുന്നത്, ഐസോമേഴ്സ്. Okay, so this isomerases helpful for the isomerization reaction, and one more is there, that is the ligases. Last one, that is ligases. Helpful for the synthesis of a large molecule. Helpful for the synthesis of a large molecule. Okay, these are the different classes of enzyme: oxidoreductase, transferases, hydrolases, lyases, isomerases and ligases. Clear? Oxidoreductase, that is helpful for the oxidation reduction reaction. Transferases, helpful for the transfer of particular group from one substrate to the other. Hydrolases, helpful for the breaking of complex substances into similar substances. Lyases, catalyze the removal of a group. And isomerases, helpful for the isomerization reaction. And ligases, that is helpful for the synthesis of a large molecule. This is about the classification of enzymes. Clear? So these are the different enzyme: oxidoreductase, transferases, hydrolases, lyases, isomerases and ligases. Okay. Next, about the properties of enzyme. The first one, optimum temperature. The enzymes are sensitive to heat. Okay, the enzymes are sensitive to heat. Many enzymes are inactive at high temperature. Inactive at high temperature, that is above 60 degrees Celsius, the enzymes are not able to work. Okay, so optimum temperature. They need one optimum temperature. Above that temperature and below that temperature, they are not able to work. Okay, so the enzyme, they are sensitive to heat. Many enzymes are inactive at high temperature, that is 60 degrees Celsius, and they become inactive at very low temperature, below the freezing point. They are not able to work. So the enzyme, they need a optimum temperature for their action. Gradual increase of temperature, the enzymatic activity is regained. So the gradual increase of temperature, okay, that helps to regain the enzymatic activity, and this property that help the different material, meat, vegetables and all that we keep in our refrigerator to be like that without any damage. That is the gradual increase of temperature, and that time the enzymatic activity is regained. So the enzymes will act only on a optimum temperature above 60 degrees Celsius, that is the high temperature, and below the freezing point the enzymes are not able to act. Okay, that is the optimum temperature. Then specificity. They are highly specific in their nature. What is meant by specificity? Each enzyme can act only on a specific substrate. First ഞാൻ പറഞ്ഞു enzyme act ചെയ്യുന്ന എന്തിലാണ്? സബ്സ്ട്രേറ്റിലാണ്. ഓരോ എൻസൈമും കറക്റ്റ് ആയിട്ടുള്ള സബ്സ്ട്രേറ്റിൽ മാത്രമേ ആക്ട് ചെയ്യത്തുള്ളൂ. This is called the enzyme specificity. Okay, so the highly specific, the enzymes, they are highly specific, and each enzyme can act only upon a specific substrate. Very important term, specific. They are specific in their substrate. ഇഷ്ടമുള്ള സബ്സ്ട്രേറ്റുമായിട്ട് join ചെയ്യില്ല. Specific substrate, and this is called the enzyme specificity. So next one, that is optimum pH. So first we studied optimum temperature, next specificity, and third property, that is optimum pH. So this enzyme, they are highly active at specific pH. Temperature in the case നമ്മൾ പറഞ്ഞു. Optimum temperature is needed. High temperature and the low temperature, the enzymes are not able to act. So here also the enzyme activity decreases above the pH, or the pH is more, or below the pH, or more pH the enzyme activity decreases, or less pH the enzyme activity decreases. So a specific pH or optimum pH, okay, use that word, optimum pH. the highly active at specific pH. So below if the pH is small, the enzymes are not able to act. If the pH is less, the enzymes are not able to act. The enzyme activity decreases. So that when the enzymes activity work, or when the enzyme work properly, the highly active at a specific pH. Next about enzyme substrate complex. First itself I told the enzyme that act on a product, that is called the substrate. The enzyme that join with the substrate to form enzyme substrate complex. And the speed of the chemical reaction is determined by the enzyme substrate complex. And the last one, effect of substrate concentration. The effect of substrate concentration. Velocity of a reaction increases with increase in substrate concentration. If the substrate concentration increases, the velocity of the reaction increases. And if the substrate concentration continues to increase, increase the rate of the reaction slows down. Okay, so that is the effect of substrate concentration. Velocity of the reaction increases with increase in substrate concentration. If the substrate concentration continues to increase, the rate of the reaction slows down and velocity of the reaction also slows down. These are the different properties. So first one, temperature. The enzyme, they work only on a specific temperature. If the temperature is above the optimum temperature or below that, the enzymes are not able to act. Second one, specificity. The enzyme that act on a specific substrate, that is called the enzyme specificity. Third one, optimum pH. If the pH is less or the pH is small, the enzymatic activity decreases. And next one, the enzyme substrate complex. The enzyme combines with substrate to form enzyme substrate complex. And last one, that is the effect of substrate concentration. If the substrate concentration is more, the velocity of the reaction is also more. And if the substrate reaction continues to increase, the rate of the reaction slows down. Okay, so this is about the properties of enzymes. Okay, clear about the properties? Okay. Next about the mode of enzyme action. So I told the enzyme that act on the substrate. And the enzyme, there is a binding site. Okay, so enzyme, there is a binding site for the substrate. E, that is enzyme. S, that is substrate. A specific binding site for the substrate. Okay, and form the enzyme substrate complex. Okay, like our lock and key. Okay, so we call it as lock and key method. The combination of enzyme with substrate to form enzyme substrate complex. Okay, so this is the enzyme substrate complex. And if needed, the enzyme is and substrate, they get dissociated. So the enzyme, they join together to form enzyme substrate complex. There is an active site in the enzyme that help the substrate to bind with it. There is an active site in the enzyme to bind with the substrate and form enzyme substrate complex. Okay, and this enzyme substrate complex, they dissociate and form enzymes and separate the substrate. And we call it as lock and key method. That is the mode of enzyme action. And there are certain substances that inhibit the activity of the enzyme, and that are called enzyme inhibitors. Certain substances that inhibit, that prevent the action of enzymes, and we call it as the enzyme inhibitors, and that process, that is called the enzyme inhibition. Okay, so that is all about enzymes. So we studied what are enzymes. Enzymes are biological catalysts. Enzymes are biological catalysts. They speed up the chemical reactions. And we studied the classification of enzyme, different classification, oxidoreductase, transferases, lyases, isomerases, ligases, etc. Next, we finish the properties of enzyme. They work on optimum temperature, enzyme specific. The enzymes are specific in their nature. Optimum pH is needed. Enzyme substrate complex and substrate concentration. That is all other different properties of enzyme. And this is the mode of enzyme action. How the enzyme act. The enzyme, they act on the substrate. In the case of the enzyme, there is a binding site for the binding of the substrate and form an enzyme substrate complex. The substances that inhibit the action of enzymes are called enzyme inhibitors, and that process is called enzyme inhibition. Okay, and next about another two small topic, that is first one, hormones. That you know. What are hormones? Hormones are chemical substances that is needed for the proper functioning of the body. Hormones are chemical substances and these are released from the endocrine glands. What is the another name of endocrine gland? Ductless glands. And these hormones are not able to act on the place where they are released. They act on the place where they are needed. And that place where the hormones are needed are called the target sites. So hormones are chemical substances needed for the proper functioning of the body. Every function, all the functions of our body, that is controlled by the action of hormones. And in case of plants, there is no specific endocrine glands. There is no endocrine glands that you know. So the phytohormone, last chapter we studied auxin, gibberellin, cytokinin, abscisic acid. These all are the growth hormone. These are the phytohormones or the plant hormones. And our last section, that is vitamins. That compounds needed for the proper functioning of the body, that is vitamins. Okay, and our body, they need the vitamins in a optimum quantity. Below that level, it causes different types of deficiency diseases. Okay, that all you study. So there are two types of vitamin, fat soluble and water soluble vitamins. Clear about hormones? Hormones are chemical substances that is released in our body, and these hormones are released from the endocrine glands. And in case of plants, there is no endocrine glands, so the hormones, they are released from the plant body itself. They are called the phytohormone. And vitamins, they are also needed for the proper working of the body. And the vitamins also, they need in a specific correct quantity. Below that, it causes different types of deficiency diseases. And the vitamins are of two types. They are fat soluble and water soluble vitamins. Okay, so clear about enzymes. Enzymes, their action, properties and all, and vitamins and hormones. Okay. That is all about this chapter, and I will give you the notes. Please study that notes only for this chapter. Okay, and plant growth hormones also, just to copy the notes from the board. If you are not able to read or if you feel any problem while writing, call me. Okay, plant growth hormone. So the different hormones that we study, plant growth hormone, last chapter. That chapter also, please study that only. All the points I wrote there. Okay, last class. So please study that one. If you feel any difficulty, call me. So that is all about our portion. Next class onwards, I will take the revision from the first chapter if you have any. That is mainly through the live class. So all must present and all must log in with your own ID, and otherwise I am not able to accept you. That is why. So please study properly. Thank you. Have a nice day.